is a lot younger.
It's kind of incredible to think about life on that scale.
Mm-hmm. Yeah, and every animal grows differently. Like, it's really interesting. Oh, I love this fish. It's so
What
cute.
kind?
Look at
Yeah,
his little
that's a
pectoral
different
fins.
fish.
It's
Those
a,
are
it's
big
a
pectoral fins.
cenomyces. Am I saying that right? I'm trying to remember how to pronounce it.
That's pretty cool. Almost like malolo. Not quite as big, but nice.
Cenomymus.
Sea star.
So I'm just like putting my own letters in the word. I love their little pectoral fins. They're really cute.
And then we haven't really reached the main coral beds yet. That's why it's kind of
I
still
think
scattered.
I think we're getting
We're
there. We're
getting
starting.
closer.
Yeah,
Yeah,
we're
it's
starting.
getting.
I would probably say this is probably the beginning of
Hmm.
the bed. Um, we're seeing a lot more
So,
diversity
diversity for sure. Yeah.
for sure. It's going to be patchy. It's not going to be like perfectly planted.
Hmm.
You're going to have little patches where there isn't anything growing, and then like some patches
Yeah.
that are really
Lots.
dense. The deep sea is known to be patchy in this way. The reason why this area is particularly interesting is it's not as it's so dense and less patchy than we've normally
Hmm. Hmm.
experienced, um, and that's likely because of the high current rates and being
Lots of
so
food.
close to the island. Yeah. There's some cookie star, and we're seeing a lot more coral.
Yeah, a lot more than when we first
Hmm.
started.
Yeah. When we first got to the bottom, I was like, yeah, this is what I expected. And then as we went on, I was like, wait, I thought it was gonna start sooner.
Well, and at least now we got more information on where does it start to pick up
Yeah,
and
where does it
where the
start?
boundaries are. Yeah.
Hmm.
So the white coral, is that not doing so well, or what's going on there? Is that what it's meant to look like?
Um, so when it's white like that, well, it depends. Some of the corals are doing just fine, and some of the corals are dead. The hemicorallium, it often grows where it's really pink near the middle, and it just naturally has like white tips. So if you see a really like pristine looking colony that isn't broken in any way, then it's pretty healthy. When it's dying off a little bit, you'll notice it starts to break up a bit, uh, and you'll have it be broken. It doesn't mean it's dead. It doesn't mean it can't recover, but yeah, like this one coming up into the middle of the screen, I would say that's a pretty healthy looking colony, but say the one, the next one up in line is not as healthy as that first one. But, you know, these animals live long lives, like I said, and you'll notice like tissue receding in certain areas. This could be for a number of different reasons. There are sea stars that do eat the coral, and we will likely see some of those around here, and they'll come, oh, there's one. Um, they'll come around and they'll just gnaw 'em away, and that's why you'll see a lot of bear skeletons. So there are coral predators.
Predators.
Um, it could be uh, a fish swam through this coral. These corals are very fragile. They can be broken very easily, so you'll notice broken parts that way. Or it could be natural death. There's a lot of reasons why we see the corals in the shapes that they are. And they can also grow in different shapes based on uh, the overall current over time. Uh, say they end up growing next to another really large colony, they might change their shape to better feed in that area and take advantage of the space where they've settled. They can't get up and move, so you'll notice that they'll change in order to better feed in that area and survive. So this is a Acanella, a bamboo coral coming up, and it's got a bunch of snake stars in its branches. The snake stars tend to be commensal, um, with the corals. They just use the corals as a shelter, and they can sometimes help out with keeping a coral clean. But like this white fluffy one, that's pretty healthy, but that's a different species of Gorgonian coral, plexaurid of some kind. Oh, here's a new one.
That's a big one, yeah.
Yeah. It's fluffy. Well, it could be something like a Theorella, which is a primnoid coral. Oh, we got a little fish. That's a morid. A codling is the common name. Oh, and then you get the sea urchins will often climb, and I think they feed on the corals as well. They're often on the coral, so coral predators, those echinoderms. I hope we see some paramola. They're really fun. So these are these really big carrier crabs, and they like to carry things. They pick lots of different types of objects, usually a coral or a sponge, and they carry it around. They're very proud of their decoration. Some crabs aren't as good at choosing than others. Some have very majestic choices. So I judge them secretly in my heart.
Do we know the background behind this behavior? Is it like related to mating, or is it a camouflage type thing?
I think it's more camouflage. They're like, hey, I'm a coral.
Eat this, don't eat me.
Yeah, but they're pretty large, so, you know, there's not a lot down here that would probably chomp on them, but a shark might. We do get big sharks down here. There's a lot of small sharks, but we could see like a hexanchus or something. Sharks do like to come by the ROV. I think they get attracted by, you know,
The electricity.
yeah, the electricity. They get curious. They're very curious animals, and they'll come by and check it out, and then they'll leave.
They probably can sense the cable, yeah, between the
Oh,
two
probably.
ROVs, yeah,
They
the
know.
high voltage.
Well, and they could probably hear it. They can feel it.
Yeah,
like
their sensors.
Have you? Well, when the ROV team turns the ROV on deck and like they turn on the hydraulics, it makes that whine. Like it's really loud. So this coral looks mostly dead and covered in other things.
So I think you might have said this earlier, but I missed it. What is that kind of tall, spindly looking coral there?
So that's a bamboo coral.
Oh, sure.
And a while back
okay.
we saw a dead one, and with all the tissue gone, it actually kind of looks like finger bones, where you have the calcium carbonate like bone part, and then there's like a cartilage protein in between, so it allows that coral to like flex and bend in the current, which is really advantageous when you live somewhere like here, where you can just be like, okay, this is fine.
Also might make it maybe a little less fragile than the other corals we've
Exactly.
been seeing. Sure.
Yes, it can bend and flex in any direction. Very advantageous to be that type of coral.
I'm kind of picturing it like those guys outside car sale.
Oh, wacky wave
sellers,
inflatable arm flailing tube man.
A lot of advantages looking like that.
Right? They are kind of the wacky wave inflatable arm flailing corals. Yeah, so these big ones, we've been calling them lepidopsis, but there's still a lot of genetic work going on with them. So there's a lot, but we keep learning. We've been learning that there's a lot more species, and it's really hard to tell them apart. And genetically, we can separate these into different clades, but morphologically, it can be very challenging to identify them. So, you know, you got to know your area and like have sampled these guys from here. But when we do see them in different areas, we like to separate them based on their morphology. If they are unbranched like this, where there isn't any branching, and then if they are branched, they're two different types.
Mm-hmm.
One type is where the branching happens at those nodes, those protein locations, and that's like the Acanella, that orange bamboo that we just saw.
Mm-hmm.
And then there's the internodal, where the branching happens on the bony carbonate area.
Mm-hmm.
And then there have been a few that kind of do both, which is weird because we used to think that it was one or the other, but we've definitely come across individuals who are kind of in between. So sort of like people, we do our own thing.
Hard to put them in a box.
Yeah, I can't put them in a box. And the more we look into it, the more we learn.
Mm-hmm. So could you talk a little bit about, like, once you get those samples from the coral and you're looking at their genetics in a lab, what is it about them that makes you say, okay, these are two different species, these are the same species?
So they look for certain markers in the genome. So there are certain little clips of the DNA that get taken out that are known to be like different between the species, and then they compare those little clips to all the other species, and the ones that are more similar get grouped together into like clades.
And do you ever have species within those clades that end up looking like totally different from one another
Oh, some of
morphologically?
them look totally different morphologically. They recently did some genetics in the Corallidae and the Paragorgia. So these corals and the bubblegum corals
That's
are
a
now
great name.
Yeah, the bubblegum. We'll probably see some bubblegums. There's some bubblegums that live over here. They're called that because the ends of the branching is sort of round and they're pink generally. And it's very bubblegummy. Like it
Cute.
just looks like the bubblegum you would get like in Halloween or in the classroom. It's like it's little bubble.
Hubba bubba.
Yeah, exactly. So apparently those two are now genetically similar and have been lumped together in the same family, which took me a while to get over.
Seems like you were really thrilled about that one.
It was a lot. It was a lot all at once because there was a whole rearranging of all the corals a while back, and so I had to relearn a lot of things, and like the things that we were calling plexurids were then paramuriciids, and now we're calling it Canthogorgidae, and now I'm confused. I'm not sure.
I know, it is interesting how fast things can change. Like, I'll have teachers and, professors at school who learn these things during their undergrad and memorize them and knew them very well, and then some new development comes and they just have to completely relearn all this stuff.
Oh, yeah. Like, the gold coral had, like, 3 other names before its current official description. So, things change a lot.
Yeah.
Some animals get described officially, and some animals, like, we're just not sure where to put them. Oh, we got a regadrella. This is a glass sponge. And then these look like dendropathes, type of black coral. It's a really fuzzy looking branching black coral. See, this one right here could be a bubblegum. So bubblegums are not always pink. Sometimes they're white. Oh, yeah. They just come in white. Some animals, they come in multiple color morphs, and they're the same species. And other animals, like if they're a different color, then they're different species. And some animals look completely different, and you're just like, okay. Yeah, you just never know. Like for a lot of animals, like color doesn't matter at all. Like it can be any color, and some like color is like your defining character. That's what makes this job hard. Is you're like, well, okay, and then especially with the black corals, those are really hard because you gotta like really zoom in and look at really small microscopic parts. So like I would love to be able to zoom in and look at everything, but we also have to get as much done as we can in the small amount of time that we've got.
So what kinds of advantages do having a more feathery look, like some of the black corals we've seen, have versus the much like harder kind of branched corals?
So the black corals have a protein-based skeleton, which tends to be
Mm-hmm.
very, very flexible. So, you know, they're finding this a solution that works in a similar way.
Mm-hmm.
You know, like you have like the hard skeleton, you have more available resources for creating a carbonate skeleton, but it can dissolve.
Mm-hmm.
You know, because if you have if you're below that carbonate, wow, that's really hard. The
I
carbonate
think would be
compensation
like with
depth,
any
Mhm.
career.
then you start to dissolve. So like that area of dissolution, then you start to dissolve, and so it can be very challenging to gather resources
Mm-hmm.
to build your skeleton in certain areas.
So as
But
sorry, you finish.
yeah, but then if you have a protein skeleton,
Mm-hmm.
that is a limiting resource in the deep sea
Mm-hmm.
because food is limiting. There's not a lot of materials to take up
Mm-hmm.
to build a protein skeleton. So it takes a lot of energy to gather what you need.
Mm-hmm. So as climate change kind of changes the levels of dissolved oxygen in the waters, how would that change that kind of pro-con balance there?
So climate change will cause a shoaling of the carbonate compensation depth. And so, you know, that makes that shallower. So dissolution will happen shallower in
Mm-hmm.
certain areas. Now, how this is going to affect the entire ocean, that's a big question.
Mm-hmm.
Um, and how things are going to affect the deep sea, it's also a big question. But overall, like if you're thinking about changes in our surface waters and how that affects surface productivity,
Mm-hmm.
that will directly affect every other ecosystem on the planet.
Mm-hmm.
Because primary productivity in the ocean does more than just feed the fish on the surface. It
Mm-hmm.
feeds everything to the seafloor, with the exception of vent areas
Mm-hmm.
where they have chemosynthesis. So those are the only areas that will be unimpacted. But it also affects us because carbon sequestration in the ocean is a big thing.
Mm-hmm.
You know, it produces oxygen. There's a lot going on with primary productivity. So changing in sea surface temperature can affect primary productivity.
Mm-hmm.
And if it's too warm, like here, you won't get mixing.
Mm-hmm.
And so in the surface waters around here, I think I was talking to you about it being oligotrophic.
Yeah.
That means there's not a lot of nutrients. But in other areas, especially in colder areas that have seasons, you'll have surface temperatures get warm and it stratifies
Mm-hmm.
by temperature. But in the winter, that stratification breaks down and there's a lot of mixing with storms and stuff, and that brings nutrients from deep down to the surface.
Mm-hmm.
So when the springtime, when the sun comes back out, there's a big plankton boom because there's a lot of food, there's a lot of light availability, and so you get a lot of primary productivity.
Mm-hmm.
We don't have that happen around here very often because it's always warm, so we
Mm-hmm.
don't get that mixing. But there are some times where we get eddies, and the eddies can bring up nutrients from deep water, and you can cause a plankton bloom and high primary productivity in certain small areas. Um, in El Nino years and La Nina years, that also affects primary productivity in the surface ocean
We
across
are
Pacific.
live.
And could you talk a little bit about what an El Nino and a La Nina are? Just like 5 second rundown,
Okay,
maybe.
yeah. So El Nino and La Nina is the sort of like shifting of weather patterns across the Pacific, and it changes. Um, Basically, it affects our weather across the whole world.
Mm-hmm.
Um, Whereas areas that are normally used to having a lot of high rain during rainy season have a really dry rainy season,
Mm-hmm.
and areas that are used to being dry have a lot of rain. And as you know, when you're not expecting certain weather, it can be very, very challenging
Mm-hmm.
to continue with your natural and normal um, plans.
Mm-hmm. And
So
if I'm
that,
not mistaken, we had one of those just this past summer, right?
hmm
Yeah.
yeah, so like we're in the middle of Pacific, so we don't feel it as much here in Hawaii, but we do feel some effects. It does affect our surf, and it does affect our rain schedule. So like we've had a really wet summer.
Mm-hmm.
We generally have more dry summers. Oh, here's my favorite. This is that fish I was talking to you about. Yeah, that one.
Yeah.
The Sladenia reminger sea butte. So they're ambush predators. They sit on the bottom.
Mm-hmm.
And what is he looking for, or she?
Uh, just
Anything.
anything that comes a little too
Anything that moves.
close. Yeah.
And what's that guy's common name?
Coffin fish.
Mm.
They're related to the monkfishes. People come up with crazy names, or frogfish. Uh, I don't know. You have to pick one. I think there's a lot of them.
Mm.
So there's a question: how do scientists keep up when it constantly changing the
Mm-hmm.
class, the taxonomy and stuff?
Oh yeah, taxonomy is always changing. Some animals are a little more stable. The fishes don't change as much, but they do change. Um, the corals are under a lot of revision, so they've been changing a lot. The sponges will change a lot.
And it's kind of just part of, I guess, with any career. Yeah, you just got to keep up with what's happening in the field.
Exactly. Yeah, just like in, say, the medical fields, you got to keep reading your
Right.
papers,
Yeah.
keeping up with the new science, because there's always new information out there. That's really important.
And as technology changes, you get better at classification, and then you get new
Exactly.
information coming in, and so.
And generally you find that, like, we've been calling something something, and it wasn't that thing all along, We're like, all right, what do we know? It's like, I thought I knew this one, but no.
Yeah.
There's another Sladenia.
Mm-hmm.
There-
So we're getting more different things.
Yeah, we're starting to see more corals. I know it kind of looks sparse to most people, but
Mm-hmm.
this is like considered dense for the
Okay.
deep sea. When we do our dives, um, I like to target, so when I plan a dive, I like to target areas that I know are going to have really cool and interesting
Uh-huh.
things. And so you might find that the more dives we go on, you're like, oh, we're finding so many interesting things. We're like, well, that was
The plan.
perfectly curated.
Yeah.
We're kind of avoiding the areas where I didn't
You
think we'd
think
find
there's
stuff,
nothing? Yeah.
right?
Yeah.
So, because we want it to be interesting. But sometimes we go to areas that aren't, we think are
Mm-hmm.
going to be interesting, there's nothing there, and that has a lot of value too. So like not finding stuff is just as important as finding
Yeah, yeah,
stuff.
because then you know that it's not there for next
Hmm
time. You won't. Then
Yeah.
how? I guess following up on the taxonomy stuff, who decides? And like, is there a large committee that has discussions, or is it like?
So it's all like peer-reviewed science. So you have experts in the field that do the work. So like Steve, he does a lot of this genetic work, and he works with other scientists who are doing another more of this genetic work, and they write papers together, and then they peer review it with other people working in similar fields. So it's a collaborative effort generally
Yeah.
on the part of the scientific community. Um, but sometimes things get published, and then the community is like, no, we don't.
Yeah.
That we
Yeah.
don't think so, and then you can revise that. Like this is a
Mm-hmm.
discussion. That's how science works. We're not going out there and just proclaiming.
Right. Right.
We're suggesting, and
Mm-hmm.
we're always open to learning
Yeah.
more.
And similar with the Hawaiian language, we see new things that we don't have words for. I wouldn't like give it a name and then that's what it is. So there's a
Yeah.
committee of
Right. We
people
want to.
much
Yeah.
smarter than me who
We need to all agree.
yeah, and work it out and come to a
Well, and if you're just one
collaborative
person
decision.
and you see something, you're like, well, I've never seen it before.
Right.
It doesn't mean no
It's
one's
brand
ever
new,
seen it
right?
ever.
Yeah.
We gotta discuss
Check with everybody.
yeah, and that's being a taxonomist and being a scientist working in this field is about is, you know, looking for these references and finding, um, you know, more information.
Yeah.
And a lot of times on,
Cross-checking.
yeah, it's like a lot of times on these cruises, we'll find new animals to science and they get described. And that's one of the exciting things we
Yeah,
do
that's cool.
here is sometimes when you tune in, you get to see something that no one has ever seen before. It's really exciting.
Yeah, that's cool.
You're like on this cusp of science.
Yep.
And then in our science chat, we'll get our, really excited scientists chiming in, be like, collect that. It's so exciting.
Yeah, that was a good question. Who decides? And it's not one person. It's a team of cross-checking and back and forth. And yeah.
Yeah, so the person who describes it does get to choose the name of the animal, but you can't name it after yourself.
Uh-huh. Okay.
But you can name it after a colleague. So that happens with a lot of species names. You'll get, names like Smithy Eye
Yeah.
and
Yeah. Yeah. I go walking in the mountains and see all these plants, and a lot of them are named after somebody
yeah.
that's not from Hawaii.
Well,
Yeah.
a lot of
Back in
the
the
plants are
days.
not from Hawaii
No, no.
too.
Back in the
Oh, back in the
the
back
native
day.
plants when
Yeah,
the botanists came through in the
yeah, and they
the
just started naming
1800s.
things.
Yeah.
Yeah. Yeah, that's hard. It's like they already had names. They were
Yeah.
perfectly good
Yep.
ones. But that is a challenge with common names too, is some some plants and animals have a lot of common
Mhm.
names,
Mhm.
and some don't have any at all. So when we try to speak in common names, especially like this deep sea stuff, it can be really challenging because some of these animals have common names that are widely accepted. Some of them have common names that all of us in the van kind of came up with. Oh, there's another octopus. Do you see the
Oh
octopus?
yeah.
Hey. Hey.
That's the second octopus we saw.
Yeah, we saw two about an hour and a half apart.
Good luck.
Hey, good luck, octopuses.
That's some of our favorite seafood.
It's so good. I could go first and taco
Dry them,
poke.
dry them. Actually brought some a mix of different dry ocean stuff.
Oh, yeah.
He'e and deer meat and some ahi and aku and
That sounds good.
shrimp and cuttlefish.
Oh,
I'll bring it
so
up
your
next time.
room is where to go. I
Yeah,
see.
I'll bring it out up on the deck maybe later on for everybody to have some.
Do you smoke and dry your stuff yourself?
Yeah, when I was getting ready to come, I threw it in the dehydrator.
Nice.
Some deer meat, made some deer jerky and some dry he'e, and we call it the Molokai Trail Mix.
Yeah.
Yeah, I think just one pilot, but maybe on the paperwork might be two names are listed, so we can rotate out. But right now we just got one person controlling the ROVs. Yeah, currently we're doing a survey of the precious coral beds outside of Makapu, and kind of we're trying to find. We started where it was kind of sparse, and those are the outer limits of the zone of lots of corals, and now we're kind of going to an area that's um, the coral beds. It has pretty good um, density and as well as different types of corals growing in the area. So kind of just doing a survey to see where the high density areas and where we started was a little low dense. So we got some bamboo coral and some pink coral and black coral, some of the gold coral and things like that. So yeah, that's what we're looking at right now.
Oh, that's the bamboo coral, the Acanella. This one. Oh, round puffy one. So all these like puffy things are sponges or demosponges. White and round. Oh, so
Megan,
like
sorry,
fuzzy?
just to
Yes?
let you know, we're getting some strong currents, so we're getting blown off. Hercules is going to be changing its heading to kind of face into it, I think.
Okay.
Um, just if you see some funniness in your video. I'll
Oh yeah, you could see how blustery it is. Look at those corals. They're like going. That's a good way to tell what a coral is if you can't tell. Like, see, these guys have rigid skeletons and they won't move. If it moves, then it can't be a hemicorallium. There's some plexaurids and yeah, some black corals. These ones are black. This like yellow one is a plexaurid. This one, that's bamboo coral.
Bamboo, yeah.
Hmm. Yeah, the fluffy one is likely a chrysogorgia. But these ones are black corals. Oh, here. It's a bit windy. Sure.
Yeah, you seeing this, Megan? All the
Yeah.
yeah, it just started ripping, and I don't know
Yeah,
if
no, it's,
if we just came over a crest or
I don't,
it just changed. I think.
it's crazy.
I don't think it's topography related. I think we.
Yeah,
Do you want me to hold the ship up for a second?
I
I mean,
don't
we're
know.
quite laid back anyway. Bridge now.
What time is it?
Hold position. Thank you. Dude, that is nuts. I mean, so the ship was moving forward that whole time, and I don't really think Atalanta's moved that much. So it's like the current has also dragged it.
I don't know who runs the currents
Yeah.
down here, but we should have, yeah,
Wow,
we
look
should
at,
talk with him, you
Kind of
know.
was in charge down there.
you can see in Atalanta cam how strong
The
that
depths
current
of
is.
Canalo.
Yeah.
We just got to the good part too. It's getting really interesting.
Yeah. Yeah. Try it out.
Pretty dense. Yeah, there's lots.
Here. So, um, sorry. So now that we're kind of focusing on this here. Dude, that is nuts. We're barely getting, and we're faced into it.
That's why there's a lot of corals here.
They're all laying down. Yep.
Oh, look at this one. There's a little sea star eating this bamboo coral. So these cookie stars are known to be coral predators, and they will just keep eating all the tissue right off until the end. Oh yeah, they're very determined.
Why are the currents so strong?
They're always strong wherever you want to go. Just seems
That's
to
usually
be
the way it is, yeah?
that's the way it is. They're like, oh, I want to go over here. It's like, oof.
The coral likes it because it's strong.
They're very adapted to living here. Got flexi skeletons. It seems to be highly variable.
Yeah, so how related is the current at the bottom to the wind and stuff on the top?
It is related, but down deeper, it's more tidally influenced.
Okay.
Right now, we're headed toward high tide. It's
And
our
it's
high
full
high.
moon, so we got
Full
a lot
moon,
of
high
movement.
high.
Yeah.
And high tide's going to be about the time we're leaving bottom.
Yes.
So that'll be when slack tide is. Which then it'll be a perfect time to be down here, but we'll have to get go home. So yeah, it's really challenging. And we were doing so well. You'll notice a lot of the corals have associate animals living in their branches. There's like crinoids and crabs and sea stars, different kinds. There's a lot of these little demisponges all over the place.
So do the corals get benefits from having those associated animals?
Some animals are beneficial. some animals are straight up eating the coral. So,
Some of them are making the coral snacks.
yes, some are having snack time.
What are some of the,
Yeah.
what are some of the benefits some of the, well, beneficial ones can offer?
So they can help like clean um, like sediment and stuff away from the coral, but generally animals that live in corals don't do much for the coral. Um, but the coral provide a lot of habitat, and so they're considered, you know, habitat builders in these deep sea environment. They make the bottom more interesting. They provide shelter, um, and a lot of animals find that beneficial. So some fishes will lay their eggs on corals. Some octopuses have been known to lay their eggs on corals. Um, some animals live their whole lives on a coral, so we'll see Metallogorgia. You'll probably see some on other dives. Um, they have like this long, thin, they're chrysogorgid with a long, thin stalk and kind of an umbrella-looking top where all the polyps are. And they always have the same snake star that lives, and they just always live together all the time. Every time we've ever seen this coral, it has the same snake star. They're always together all the time. We have no idea how they get together, but every time we see it, same. So we see that association a lot. Snake stars are very common on a lot of different types of coral species. We'll see them on coralliums. We'll see them on Paragorgia, the bumble gum coral. We'll see them on bamboo coral. We'll see them on plexaurid corals. So it's a very, very common association. We often see shrimps in corals. They like to use it. The squat lobsters, we'll see a lot of squat lobsters on corals as well. And then we'll see a lot of sea stars that just straight up eating coral. So we'll see them on each other too. But it's not as happy.
So many different species that they can support.
Yeah.
Yeah, there's a lot. We'll see a lot of polychaetes as well. Scale worms in particular tend to enjoy living on these animals.
Don't the sea stars like to climb up so that they can get access to more food that's like blown by in the current? Is that
Well, so if they're filter feeders, so like brachygids are filter feeders. They're a type of sea star.
Megan,
We
sorry
saw
to
some of
interrupt,
those.
but we've lost the plot here. So
Oh
the
no.
yeah, it's too strong. It was too strong for us to fight. So right now we're tail to tail. We can hold here until we see the current is fightable, or we could try to reposition. I don't know if it's spatially related. I think it's more time. I think just
Yeah,
at
well,
least
we're
not to the
almost at high
resolution.
tide, so
Yeah.
could have something to do with it. Yeah, if we can't move from here, we could sit down and like look at some stuff.
Yeah, we might be able to do that in the tail-to-tail configuration. But let's just, yeah, that's up to you guys, pilots. For the, if you get your, if you're happy with your delta and we're able to get Hercules down at the seafloor, we can take a look at something. But it's going to be pretty bouncy, actually.
Yeah.
You know, because I don't really know if that's gonna probably stay away from the seafloor. Yeah. So it's more, it seems to me like a time thing, right? We could just sit here and wait until the current
Right.
stops, or
I don't know if it'll stop.
like diminish. It came up like out of nowhere. You know?
Yeah, maybe it could stop. I have no idea.
Yeah.
We haven't been able to really like nail down what this area is like.
Well, there could be internal waves in this area too.
Oh
that
yeah,
are
probably. Well, I don't mind waiting.
Your auto head is on.
Like, could we just tow a little bit, or?
We could tow, but I don't. It's kind of. I think it's. Probably. I don't think we could tow to a place where the spatial extent would have been a different current. I think
Okay.
it's probably just the time right now. So we
Right.
could either try to wait it out. Yeah. Yeah, there's not a lot of time. So I don't know how much we have besides just waiting.
Okay. Well, you know, waiting is the game.
Yeah.
I don't want to say like, oh no, let's just go home.
No, yeah. I mean, we'll just watch that bounce there with oh, you're 7 meters off. You're fine. Yeah, yeah.
Yeah, it is pretty bouncy.
I think so. Atalanta's camera is facing down, and we can see the current there. So maybe if we see that that stops going so fast, we can start to try to regain control, but we're kind of helpless at the moment.
Hmm.
Helpless. Yeah, it'll be that way until we get out of the current, which will be... Well, you could put some backwards way on it, yeah? Or are you worried about your comp right now, your res? Because you could, yeah, put some, try to back up and see if it at least alleviates some of that. I think it'll stop. Yeah, could do. Yeah, that'll stop with the up and down yanking at least. Yeah, I mean, if we're just here waiting anyway, might as well. So you can, if you want to. Yeah, that's fine. As long as you don't see, you're not heaving into it, I think we'll be okay. Yeah, you got a high enough altitude. I think we're okay. Yeah. Yeah, I mean, since we're stretched out, you could pretty much go as low as you want, as long as you're comfortable with the heave in the bottom, you know, because we're not worried about the tether anymore. So there, you see that in that butt cam there. So that's Atalanta on the right. And then we can see Hercules is that bright light. Yeah.
Yeah, currently down at the bottom is a real strong current, so we had to come up off the seafloor a little bit, and hopefully it mellows out a bit. We can get back down, but we'll see. aloha kako e na hoa makamaka o ka aina. Hui kala mai ke ia manawa ano I kai ke ao ka moana, ka papaku o ka moana, no leila. Ano pi iki mako a kali ana I ke kahi wā mālie paha. Ina aole mali ina mālie maila, e iho hoa na mako I ka papaku. Ka mahalo nui au ko poe a koko a holoi mai a mako ke ia la. Thanks for everybody tuning in. We got experiencing strong current down at the bottom right now, so kind of had to move a little bit up away from the ocean floor, and we'll see if it kind of mellows out a bit, then we can go back down.
Megan, just to let you know, we are having to come off the bottom. Yeah, I think.
Okay.
Oh, I can't hear you. Sorry.
That was the end of the dive.
I think so.
Okay.
Yeah.
Okay.
All right.
Sorry.
It's okay.
Yeah.
You did your best.
Yeah. The current
The
came.
current
Yeah.
Too
is a
much.
killer over here. I know.
Yeah.
Could we fire the Niskin bottles as
All
we're
of them?
coming up?
Yeah.
Yes, please.
Pilots, do you need to be at the? Is it okay at this depth here?
Yeah, it's whatever.
Yep, that's
Okay.
fine.
We're just
Um,
going to use them
It's
as
just,
practice.
yeah.
yes. Why? What's up? Because you're pulling up. Yeah. Um, Dan, do you mind firing all the Niskins?
Sounds like he minds.
Because of the res?
Just do it. For me, Dan.
Pretty
For
Yeah.
please.
Megan. Okay.
Thank you. Okay. Yeah, whenever you want on the way up. Oh, the crafty craft.
Yeah.
Mhm. Kinda.
Pilots, I'm switching you to USB fix. See that?
I like your new golf balls. Those are pretty spiffy. Hmm. They're 3D printed. Hmm. Yeah, they got this 3D printer, and then we bought one too because we really liked it. It's a really good one. It works at sea really well.
It's on the ship.
Yeah, yeah. It's a Bamboo Labs 3D printer.
Very nice.
I feel lucky hitting that blue button. Oh, good job, Craft. See, it wanted to do it for me.
Cranking down.
Manel. I do not. Okay. Just, all right. All right, ready. 3, 2, 1, Niskin. Well, it was good practice. Was that nasal enough? Yeah, I guess so. All right. 3, 2, 1, Niskin.
Did you say NASA? I thought you said nasal. That was not NASA. No, I promise. Okay. Okay. Okay.
An,
Okay. Okay. Okay.
an
Okay.
official,
Niskin. All right. I can do NASA. All right. Hold on. I gotta prepare my voice. All right.
Of course, you know.
All right. Can't believe I did nasal for 2. All right. 3, 2, 1. Niskin.
I, I don't see it yet either.
Might have gotten swordfished.
Oh.
We got attacked by a swordfish earlier. Yeah.
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 Yeah, the swordfish, they're intense.
Let's go number 4. Yeah.
That's
3,
basically what he was saying.
2.
Yeah, you can see the
1.
vehicles in each other's
Niskin.
cameras too.
Gosh darn.
Boy, who rigged these things?
Well, a swordfish might have unrigged them.
Ah,
I don't know. If that's possible. We'll go with it.
yeah, those pesky sharks.
All right. 3, 2, 1. Niskin.
I thought I saw it move, but it hasn't tripped.
Oh.
So this is, take note of the blue here, the meters. So
Oh, that's okay.
just to confirm, we've tried 3, 4, 5 thus far, but not 1 and 2 yet.
We did
No, we
also
did.
try
We
1
got
and
1
2.
and
Ah,
2.
okay,
1
I
and
must
2
have
are
missed that.
tripped,
Yeah.
but
Dang. I think they did too. These guys
Nasal.
responded to the nasal. All right, we're going back to nasal for 6. All right. I don't know if I am. 3, 2, 1. Niskin. Jesus Christ. No. At least we know I can't be blamed. Oh.
yeah. What do you need the knife for?
Dan, I'm currently still holding station because I know you want to try to take that tether wrap out. Go ahead. Roger that. Thank you. Yep.
Well, we do want that to work. We gotta suction all those animals. I'm gonna inquire again about the fish sampler. Apparently, Falcor has a fish sampler we can print. I don't know, they said they caught a bunch of fish with it. Yeah. But if they can suction fish, I want to suction a fish.
Don't they also have a net that they
Oh.
were using?
Oh, maybe, I don't know.
I heard they were catching fish with a net too.
That would be impressive. One of the old sub-pilots, he caught a fish with the manipulator. Super, like, Miyagi style.
Kung Fu Panda.
Yeah.
Okay, thank you, Bridge.
Control van, this is the bridge. Comms check.
Loud and clear, Captain.
No.
Oh, little fishies.
Oh, look at those fish.
Oh,
They're
yeah.
so
Little
cute.
Just little friends.
They're like, I'm just going to associate with you.
Such a pretty color.
Yeah, fish will, like
they looking
anything floating in the water, like these little fish will come and associate with it as like a shelter. And so, like any floating object will have like these full communities of fish and things. And the bigger fish will come and like feed at these places.
I think
So every time like we recover, especially during the daytime, you'll see this. Like little things following us. At nighttime, you'll get lots of squids. Squids are fun. They ink you. So you'll see all their like pseudomorphs and they're just like ink all over the place.
Is there any potential of that like messing up the cameras or no?
It generally doesn't stick to the camera.
Mm-hmm.
So,
Fair enough.
yeah.
I think I saw little fish like these, like those at a buoy earlier this week.
The ink will stick to other parts of the vehicle though. Like, so the ROV guys don't really like it.
Yeah.
They're the ones who have to like clean it off.
Yeah.
You love the squid ink? Okay, I'll let the squids know.
More, please.
Pretty sure I heard him complain about it once.
I wouldn't be surprised.
So for the ADCP at the surface, it's reading half a knot that we're facing, ships facing into it. So at least at the surface it's half. I don't know what it is where you're at there, but we could try to come up to 50 and do it if you want. Yeah, 10 and 19.
Bridge, Nautilus deck, radio check.
Deck, bridge, loud and clear.
Alright, copy. Control van deck, radio check again.
Loud and clear, Doc. We could try it again at 50, and then if it doesn't work then, you know. We have the time because we came up a little early, so it's up to you. Not constrained by that, at least. Oh, yeah, you're right, I can see it. Look at it up. Come on. Oh, yeah. Okay. Yeah, all good. Roger. Bridge, nav. Can we track a line forward at the ship's current heading, speed 0.3 knots? Thank you.
Currently, I'm secondum.
Okay. Yeah, Dan, they didn't zero on the payout for when we went over when we were at the surface. I
Oh, you
noticed
gotta
that the payout
select
was
an
way wrong.
expedition,
So
and then, oh, it should be up here. That's really strange. You're logged in. You're active on this expedition. Your access to this cruise is pending approval. That's why. You have to wait to whoever needs to approve it. I don't know who that
what do you
is,
need,
but
Dan?
maybe ask Rachel. Yeah, once you're approved,
Yep.
you'll have a chat tab, a Grafana tab, and everything.
Roger. Deck control.
Yeah. Like if you want to participate in any of these other ones from
Just letting you know
shore,
we are all stopped at
definitely
50
like sign up
meters. The winch
beforehand
payout
so
is a
there's
bit
more
off
time.
from our depth, so we'd like to zero that at the surface if you can. The ship
Yeah.
is streaming forward at
Like, it
0.3
could be based on your
knots, and
email, like you
we're
have multiple
ready to
emails.
recover.
Yeah. I know, mine used to come to all three of my emails. It was getting really confusing.
Hey, just a reminder to the back row that we need all quiet for operations, please. Thank you.
Hey, what's up?
Quickest ascent ever.
meters, copy. All stations have a visual on Atalanta.
Copy.
Copy.
All stations have a visual on Herc. It's between the goalpost after the vessel.
Copy. Copy.
Copy.
I'll stop in the winch, Robert, if you want to zero there.
Zeroed.
Okay, continue to come up. Control van, can Herc give a pull ahead there?
Copy.
Okay, Herc, that's all good. Thank you.
Roger.
Control van, deck, how's your visual on Herc?
All good, thank you.
Water. All right, we're just going to hold it here, wait for the daisy chain, and then we'll have a look at the recovery line.
Copy.
stations, that's the recovery line connected, starting to recover Herc.
Copy.
Copy.
Okay, we're all stopped on the crane there, just to give Herc a chance there to come over to our port side.
Copy that, working on it.
Copy that. If you need us slacking out a bit, let us know.
We're okay, thanks.
That's Herc, 2 meters off the port transom.
Copy.
That's Herc, clear the transom, coming under the crane.
Copy.
Herc out of the water.
Copy.
Copies.
Voltage is secured.
That high voltage secure.